Welcome to October's MycoTalks. I'm Gordon Brown from the MRC Centre for Medical Mycology at the University of Exeter, and my co-chair today is Geraldine Butler from the University of College in Dublin. So as usual, we have two really excellent speakers lined up for you today. The first is Bernd Schnabl from the University of California, San Diego, who's going to tell us about the gut microbiome and liver disease. And the second talk is from Michal Olszewski from the University of Michigan, who's going to talk to us about immunomodulation and meningoencephalitis. Now, if you haven't joined us before, the purpose of this webinar series is to provide the community with an open access platform in which we can share on the diversity of all the fantastic research that we all do that contributes to the broader field of medical mycology. So we have two talks every session, each aimed to cover a slightly different area of our field, and each talk will last about half an hour. And at the end, there will be a joint question and answer session in which we will field questions to our speakers. And as you'll see at the bottom of your screen, there's a Q&A button. Please use that, not the chat function, to submit questions. And Geraldine and I will go through those at the end of the session. You won't see those questions. They won't be shared with the audience, but they will show up on our screens. So we record all our talks. So if you know of a colleague, friend, or student who's missing this talk, who should watch it, please encourage them to come and visit our YouTube channel on the MRC webpage, where all of the talks are posted after they occur, normally about a week later. So just to highlight our upcoming MycoTalks, so we have one on November the 20th coming up. This is slightly earlier than our normal schedule. This is to miss the American Thanksgiving. We're skipping December, and then the next MycoTalk will be in January, back in the normal last week of the month slot. So please look at those. And just to remind you, please, to register for each MycoTalk, and you can do that through the link, as you can see at the bottom of the page here on our CMM website, if you can't write that down quick enough. And then also just to draw your attention to the upcoming MycoClinics seminar series. So this is a quarterly series, which is very clinically focused on presenting clinical case reports, and there's one scheduled now for the 11th of December. So I would encourage you to attend those. So with that, I'll stop sharing now, and it's my pleasure really to introduce our first speaker, and that's Professor Bernd Schnabl, who is the founder and current director of the San Diego Digestive Diseases Research Center, based at the University of California, San Diego, as I've already said. So Bernd is a clinician scientist specializing in gastroenterology, who's published extensively in this field, over 270 papers, and is the recipient of many awards, including an NIH Merit Award. Now, his research interests all focus around the liver, and his focus really in three areas. The first is on the contribution of microbial products and metabolites to liver disease. The second area is genetic and molecular mechanisms of chronic liver disease progression. And the third area, which is what really focused on medical mycology to some extent, is the intestinal microbiome, metagenome, and transcription, and how it's associated with liver disease. Bernd has had a very strong interest in the microbiome in recent years, and he's made major contributions to our field in this area. And Bernd, over to you. We look forward to hearing about your latest research on the microbiome and liver disease.
Yep, thank you so much, Gordon, for introducing me. Thank you so much, Geraldine, also for inviting me. Let me share my slides. It's really a great honor to be in this MycoTalk. I will talk about, like Gordon said, about the gut microbiome as therapeutic target for in liver diseases. These are my disclosures. And this is my outline. So I will introduce initially the alcohol-associated liver disease and hepatitis to you, and then I will also give you a little bit introduction about why we are studying, in fact, the gut microbiome or microbiome in the context of liver diseases. I then want to switch over to our studies that we have done in the past about showing you how the composition of the gut microbiome and microbiome changes in patients with alcohol-associated hepatitis. And then I want to really highlight like three mechanisms that we have investigated over the last probably 10 years, and how we believe the gut microbiome, so fungi, contribute to alcohol-associated liver disease. So let's start with the natural history of alcohol-associated liver disease. If somebody drinks chronically alcohol, has an alcohol use disorder, they all will end up with a hepatic steatosis. Their liver gets larger, and you can see here, this is an H&E slide, the parenchymal cells in the liver, also called hepatocytes, they have these lipid droplets, these white droplets here, and this indicates, this hepatic steatosis. Then probably less than 50% of the patients, despite continuous drinking, actually progress to more advanced liver disease. They develop liver inflammation, also called steatohepatitis. And then in more advanced liver diseases, we have now the development of liver fibrosis. So the parenchymal cells, the hepatocytes, are replaced by the scarring tissue shown here in this blue staining. And then in further advanced liver disease, patients develop cirrhosis. So this is essentially an end-stage liver disease, typically requires liver transplantation. And you have now this, more or less, these nodules forming of scarring tissue around still some of the hepatocytes that are still present. But this is a liver that does not completely function any longer. And then some of these patients then, on the basis of this cirrhosis, develop hepatocellular carcinoma. I will also talk a little bit about a disease which is called alcohol-associated hepatitis. As you can see, it can arise on any stage of liver disease in patients with alcohol use disorder, but predominantly this occurs in patients with chronic liver disease, so with cirrhosis, and we also call it acute and chronic liver failure. Alcohol-associated hepatitis is characterized by this rapid onset of jaundice, so the bilirubin is very high, between 10 to 20. These patients actually have a very high mortality within 90 days, between 20 to 50%, and we essentially have no effective therapy. This is why the reason is why we are so focusing on this disease and looking for etiologies and pathogenesis. How is alcohol metabolized? So once somebody drinks alcohol, most of the alcohol is already absorbed in the stomach and the proximal small intestine, and then it goes via the portal vein directly to the liver. So the liver is essentially the organ that metabolizes most of the alcohol in our body. We have two main enzymes that metabolize the alcohol. One is the alcohol dehydrogenase. The second one is a cytochrome P450 2E1. And both metabolize ethanol into acetaldehyde, which is very volatile and reactive. And acetaldehyde can create reactive oxygen species, can cause lipid peroxidation, ER stress, and that leads to hepatocyte injury. Now, you might wonder yourself, well, you just told us that 50% of the patients do not get advanced liver disease despite drinking. So why is this? We know now risk factors that are associated with the progression of alcohol-associated liver disease, and these include like comorbidities like hepatitis C infection, HIV, also obesity and the metabolic syndrome. We know that the drinking pattern actually plays an important role, the amount, obviously, duration, and the type. We have learned probably over the last decade, for example, that binge drinking is a very harmful way of drinking alcohol, and that is associated with progression of liver disease. And also drinking outside meals is more harmful or more riskier to develop alcohol-associated liver disease in advanced stages as compared to drinking a glass of wine with dinner. Dietary factors, especially the synergistic effect of Western diet with alcohol, is important. And then we have some genetic factors, gender and ethnicity. And what I want to convince you today is that probably the intestinal microbiome is also a very important factor and risk factor for progression of alcohol-associated liver disease. So why do we talk about microbiome in the context of liver diseases? Well, this is due to this to the direct communication between the gut and the liver. As you know, the portal vein essentially drains all the blood from the gut to the liver. This is very important during health. Then we have all the nutrients are being absorbed. They go to the liver as a fourth organ in our body. These nutrients are being either stored or metabolized. But if there is a problem in the gut, and many of these patients, they have this, what we call a leaky gut, so increased intestinal permeability. And in this case now, LPS, but also other microbial-associated molecular patterns now translocate from the gut to the liver. And here they can act more or less as a second hit. So you have this onset of liver disease through the alcohol, and now you get this leaky gut. LPS goes there, and then you can get a progression of liver disease. And then the liver talks directly back to the gut via the bile ducts and the bile, especially bile acids, which have an important function and have an important role in the composition, or changing the composition of the gut microbiome. So in the following, I just want to briefly show you some of the changes that occur in patients with alcohol-associated liver disease with respect to the bacterial and fungal microbiome. I restrict myself to just one slide about the bacterial microbiome. We have characterized this like a long time ago. We did sequencing, metagenomic sequencing from patients with either healthy controls or patients with alcohol use disorder, shown in green, or patients with alcohol-associated hepatitis, shown in orange. And there's a clear change or differences in the beta diversity based on this PCA analysis. And many of the pathobionts that we know, like Enterococcus faecalis, but also E. coli, are dramatically increased in patients with alcohol-associated hepatitis as compared to controls and alcohol use disorder. Now, switching over to the fungal component of the microbiome, and how did we get into this and study this? And this experiment I'm showing you here is quite old. So we have probably like 10 years ago made this discovery that in mice that are fed ethanol, and we feed these mice a liquid diet. So the liquid diet contains everything, the carbohydrates, the lipids, and the proteins. And then we typically mix into this liquid diet, we mix the ethanol. Otherwise, the mice would smell the ethanol and then they would not drink it. And once we fed this ethanol once we fed this liquid diet containing ethanol, all of a sudden what we saw is that we can actually culture fungi and yeast from the stool, from the feces from these mice. And as you know, in control mice, we hardly can culture any fungi. In this case, these mice also received the entire same liquid diet, except obviously the ethanol. The ethanol was replaced by isocalorically with glucose. So some of the mice show some colonies of fungi, but most of them are also negative. And this was an eye-opener for us because this was also a black and white experiment and result. So based on this, we started actually our journey to dive into the microbiome a little bit more. Initially, we characterized also the fungal component by direct culturing of stool from patients with these alcohol use disorder in yellow. So these are the more milder liver disease. And then these patients with alcohol-associated hepatitis, this very severe liver disease. And you can see that we can also culture many more fungi from the patients with alcohol-associated hepatitis. And when we did qPCR, single colony PCR, we can also confirm that Candida albicans was also significantly increased in patients with alcohol-associated hepatitis. We further confirmed this by culture-independent ITS sequencing, and you can also see that Candida species, shown in red here, take over essentially in patients with alcohol use disorder and alcohol-associated hepatitis as compared with the controls. Here we have about 25% of the patients of the microbiome are Candida species. The next question that we asked ourselves was also, is this a reversible or really irreversible process? So to answer these questions, we actually collected stool from about 60 patients who were actively drinking. And then 2 weeks after abstinence, we also collected from the same patients, so paired stool samples, 2 weeks after abstinence from the same patients. And then we again did ITS sequencing. And as you can see here, 2 weeks of abstinence already is enough to get you a significant difference in the fungal component or fungal composition in this patient cohort. And when we look at Candida albicans specifically, we see a significant decrease of Candida albicans after only 2 weeks of abstinence. And furthermore, we also looked at anti-Candida albicans specific IgG antibodies in the serum. And even now, 2 weeks of abstinence was sufficient to also get the IgG levels slightly decreased in this patient population. So fungal dysbiosis in this patient cohort is not completely irreversible. At least we get partial reversibility, and this is happening very fast, like after 2 weeks. We haven't done longer experiments and looking, would we get a complete reversal to healthy normal controls? But that would certainly be a very interesting experiment. So in the following, I want to show you now 3 mechanisms how we have determined how the gut microbiome actually contributes to experimental ethanol-induced liver disease. And the first experiment, or the first mechanism that I will describe to you, is via pattern recognition receptors. And at the very beginning, when we saw this dramatic increase of fungi, of culturable fungi in the feces from mice that were treated with ethanol, we initially obviously asked, is there overall a contribution of this fungi to alcohol-associated liver disease? So the experiment that we set up is essentially we gave the mice, the control mice, we gave them vehicle, and then we treated a subset of these mice with amphotericin B orally. Amphotericin B, as you know, if you give it orally, it's gut restricted and it's not being absorbed. And as you can see here, in the total fungi, and this is done by qPCR, also increases in the culture-independent PCR technique. And then when we use amphotericin B, we can very nicely decrease the fungal burden back more or less to baseline from control-treated animals. I have also mentioned to you that the patients and also mice with ethanol-induced liver disease, they have this leaky gut. So after this ethanol feeding, we see a translocation of beta-glucan into the systemic circulation in mice that were fed ethanol as compared to the vehicle-treated or in control-fed mice. And also beta-glucan is also now decreased with treatment of amphotericin B. So this translocation of this fungal products to the systemic circulation is also decreased. Now, obviously, the most important question for us was, is liver disease also suppressed in these mice? And yes, it is. So we have 2 primary endpoints that we are looking at mostly. One is the ALT, which is a specific marker for liver injury in this mice. It goes up after ethanol feeding. With amphotericin B, we can see a very nice decrease in liver injury. And we can see also that that this steatosis that I described to you, the second hallmark of alcohol-associated liver disease, is also significantly decreased. And another important feature is liver inflammation, and that is also decreased after amphotericin B treatment. And this is all published, and we did this also in collaboration with Gordon, who helped us tremendously to move our studies forward. So I'm just summarizing what we have described in this paper. What I have proposed so far is that alcohol causes fungal dysbiosis. This increases the Candida species especially. And then you can see here the intestinal epithelial cell lining is essentially the paracellular spaces are increased. We have this increased intestinal permeability, and now beta-glucan translocates from the gut via the portal vein to the liver. Here it can bind to pathogen recognition receptors, and we have in the liver, we have this Kupffer cells, which are the resident macrophages. They carry now this CLEC7A or dectin-1, and via this dectin-1, beta-glucan can bind, can elicit an inflammatory response, especially via IL-1 beta. And IL-1 beta directly contributes to hepatic steatosis, but can also contribute to hepatocyte death. So this is one mechanism how fungi, through this ethanol-induced fungal overgrowth, we get increase in liver disease. A second mechanism that I would like to describe to you is via this Candida lysine, a toxin that is produced by Candida albicans. I'm sure many of you are familiar with this, and I'm not going into too much detail, but Candida lysine is transcribed from this ECE1 gene. It's translated, and then via various proteases, it's cleaved into various peptides, and one of these peptides is Candida lysine, and it is proposed to be a pore formation, to have pore formation or to cause pore formation in epithelial cells, and this also causes increased epithelial cell damage, increased LDH release into the supernatant and cytotoxicity. So we have set up an experiment, and we have done this in collaboration now with Bernd Schnabl, who helped us also tremendously moving this project forward. We used Candida lysine synthetic peptides. We incubated primary mouse hepatocytes with as a blank controls with negative controls, and then with increasing concentration of Candida lysine. And you can see that you can cause cytotoxicity and primary mouse hepatocyte death with this Candida lysine. And this increases even a little bit higher if we add ethanol to the culture of these primary mouse hepatocytes. So this was one of the experiments that we did in vitro. Obviously, the most important for us is there also contribution in vivo. And in vivo, we set up various groups of mice that we either gavaged with PBS and then fed ethanol. A second group was gavaged with Candida albicans wild type strain. A third group, we had a Candida lysine strain that was deficient in the ECE1 gene, so did not have any Candida lysine. Another group had now was gavaged with Candida albicans strain that had an ECE1 deletion, but a reintegrant of the ECE1, but lacked the Candida lysine. And last group, control group, was a Candida albicans strain that lacked ECE1, but had a complete ECE1 reintegration. So if Candida lysine now contributes to liver disease, we would expect that mice that were gavaged with Candida albicans that do not have Candida lysine, so in this case the red and the pink mice, they would have less liver disease. And exactly that's what we saw. Again, this is liver injury shown by serum ALT levels, just focusing on the ethanol-fed animals. We have a PBS group. When we feed or gavage Candida albicans repeatedly, we see an increase significantly in liver injury. And then when we gavage them with the Candida albicans that lacks Candida lysine, you see that we can reduce liver injury almost back to the baseline of PBS gavaged mice. So this was not proof, but evidence that Candida lysine probably plays an important role also in liver injury. We also saw the same effect like in hepatic steatosis and in liver inflammation. So Candida lysine really exacerbates ethanol-induced liver disease. And coming back to this cartoon here, we propose that Candida lysine is produced by Candida albicans in the intestine. It then probably can also translocate via this increased intestinal permeability to the liver, and here it can now directly cause hepatocyte damage. We are still working on Candida lysine, and we do this now also again with help from Bernd Schnabl and Salome Leibengut-Landmann. So in this case, and these are a couple of unpublished studies, we also asked ourselves, do these Candida albicans strains that we can isolate from our patients, do they have different cytotoxic profiles? So from the patients who were Candida albicans positive, we isolated Candida albicans strains, and then we co-cultured them for 24 hours with CACO2 cells, so epithelial cell, colon cancer cell line, and then we measured the LDH toxicity. We used the Candida albicans wild type strain, shown in orange here, as a positive control, and then one Candida albicans strain that lacked an ECE1 gene as a negative strain. And you can see that we can also about 30% of the CACO2 cells die after incubation with the Candida albicans strain, and none with the Candida albicans strain that lacks Candida lysine. And then you can very nicely see that we have different cytotoxic activity from the Candida albicans strains from our different patients. So one column essentially represents one patient, and we have patients where we do not have, or where the Candida albicans strains do not elicit any cytotoxicity. When we use an optimal cutoff, we can also very nicely see that patients who have low toxic cytotoxic effect of Candida albicans strains that we isolated from their stool have a better survival curve than patients with a high cytotoxic effect of Candida albicans. So this is, like I said, probably the second mechanism. Candida lysine from Candida albicans contributes to cytotoxicity of primary mouse hepatocytes. And finally, in my last few minutes, I want to show you also another and a third mechanism how we believe that gut fungi, and especially Candida albicans, contributes to experimental ethanol-induced liver disease. And this is via the Th17 cells and IL-17 production. And we got very inspired by this paper from Petra Bacher. Many of you probably know this. She showed that Candida albicans triggers the or primes Th17 cells in the gut. They then go into the systemic circulation, including into the tissue from our lungs, and then up on inhalation of fungi antigens. And in this case, not specifically Candida albicans, but also like Aspergillus and other fungi, they can now react. They are cross-reactive now. And in patients with chronic airway disease, they can elicit this pathologic Th17 response. So when I read this paper, I thought, wow, this could happen very similar in our system. In our system, obviously, we would not inhale the antigens, but we would, what we hypothesized is, again, that we would have Th17 cells primed in the intestine. They would migrate now to the liver. And now, again, we have this via this portal vein, we have this direct connection of the gut with the liver. And via this paracellular increased permeability, fungal antigens can now re-stimulate and activate Th17 cells in the liver, and they can probably contribute to liver disease. So that's what we were set out to prove or not prove. In this case, we started our experiments, and this is also in collaboration with Petra. Actually, she did all of the human experiments. We used PBMCs from patients with alcohol use disorder and from non-alcoholic controls. We stimulated them with Candida albicans extracts, and you can see that we have significantly more IL-17 positive activated CD4 T cells in patients with alcohol use disorder as compared to the non-alcoholic controls. Petra also used Saccharomyces cerevisiae as a control peptide, and there was no significant difference between these between the two groups. We further characterized the response, and it's a classical Th17 response, so with increased IL-22 and increased IL-17. The question now for us was, yes, we have these Candida albicans specific Th17 cells in the periphery of these patients. Are they also in the liver? And potentially, do they show the same TCR, so T cell receptor? And in order to do that, what Petra did is she used, again, PBMCs. She incubated them with Candida albicans lysates, and then she extracted the Candida albicans reactive T cells, and then did either single-cell RNA sequencing, or more importantly for us here was the single-cell T cell receptor sequencing. We did the same thing from a liver biopsy from the same patients. So these were paired samples. And in this case, because the liver biopsy is a tiny little thing in patients with, when they undergo this liver biopsy, so we could not get enough, or we could not enrich enough T cells from there. So in this case, we did bulk TCR sequencing, and then we essentially looked for bulk and single-cell TCR overlap between the PBMCs and the liver biopsy. And indeed, what we found is these are two patients that we did. We actually had a increased TCR alpha and beta chain representation in the liver as compared with the PBMCs. And when we looked at the UMAP from the single cell TCR sequencing and single cell RNA sequencing from the PBMCs, we overlaid, or Petra overlaid, the cells that we could also found with the same TCRs in the liver. And you can see that we have a major proportion of these cells are actually Th17 cells. But however, we also have Th1 cells here, and this is quantitated over here. So suggesting that indeed we have the same clonal Th17 cells that the Candida albicans specific in the liver as opposed into the and in the PBMCs, so in the systemic blood. But to really show that now these Candida albicans reactive Th17 cells are really primed in the intestine, we actually in our case had to turn to a mouse model. And in this case, we used a photoconvertible KD mouse. So in this case, the KD mouse has a green, or every cell is green fluorescent, and up on photoconversion with a laser of 4 or 5 nanometer, you can turn now irreversibly the cells into a red state, and then you can track these cells throughout the body. And we obviously focused on the liver. So here we looked for red cells that produced IL-17 up on Candida albicans stimulation. And indeed, what we found is that once we looked for these red IL-17 positive cells in the liver, we found more in the ethanol-fed mice as compared with the control mice, suggesting that indeed these photoconverted mice that we specifically photoconverted in the intestine, and specifically in the mesenteric lymph nodes, they come from the intestine and go to the liver. Now, to really show that these mice that these Candida albicans reactive T cells indeed contribute to liver disease, we had to do an adoptive transfer. We used reporter IL-17 GFP reporter mice. We primed them either with Candida albicans or with Saccharomyces cerevisiae as a control. We isolated CD4 T cells from the spleen, co-cultured them with DCs, and stimulated them with Candida albicans lysates or Saccharomyces cerevisiae lysates. And then we injected these Candida albicans primed T cells or the control primed T cells into the mouse and fed these mice chronically alcohol. And the results are shown here. We get a quite good engraftment of these Th17 cells, and we get significantly more liver injury after adoptive transfer of Candida albicans primed Th17 cells as compared with the Saccharomyces cerevisiae primed Th17 cells, and we get more hepatic steatosis. So these are my key takeaways. I hopefully have been able to convince you today that liver disease is associated with changes in the gut microbiome. I've shown you 3 mechanisms. One is fungal glucan, beta-glucan, and Candida lysins promote alcohol-associated liver disease. And the third mechanism is the Candida albicans specific Th17 response contributes to alcohol-associated liver disease. I would like to thank my entire laboratory. Many of these folks have actually contributed to this, but I want to especially thank everybody in the microbiome field, and I have listed them here, with whom we have collaborated over the years. Gordon, Bernard, Petra, and Salome. They really welcomed me into this fungal community, and I'm very grateful. I'm a physician scientist, I'm a gastroenterologist, I'm not a mycologist, but getting their expertise and their tools and their knowledge, I think, helped us dramatically moving this forward. And without them, we could not have done all of this. So thank you so much for your attention. I'm looking forward to the questions at the end.
Thank you very much, Bernd. That was great. Can I just remind our audience to direct the questions in the Q&A, and please proceed them with the name or the initials of the speakers so we know who the questions are directed at. So I'm delighted to introduce our second speaker, Michal Olszewski, who's a professor at the University of Michigan Medical School and also a research scientist at the Department of Veteran Affairs. but most of you might know him best from his role as the Mycology Section Editor for PLOS Pathogens. So Michal did his PhD training at Michigan State University and then postdoc at the University of Michigan, which is something he shares with me. But he works on Cryptococcus neoformans and particularly the host defense in the infected brain. So we're very much looking forward to hearing your presentation.
Okay, I think everybody can hear me and see it. So today I will tell you about immunomodulation in cryptococcal meningoencephalitis and our favorite motif in the crypto field are Star Wars. So yes, there will be some digression to the lightsabers. Let me see why it doesn't advance. Okay, so we know from this quite well developed concept of the damage response framework that we can have too little or too much of the immune response, and this is particularly relevant to cryptococcal meningoencephalitis because the reason we first would get this infection in the brain is because of some defects in the immune system, but then there is also a situation where too much immune response is bad for the patients due to this tremendously damaging effect of the immune cells in the brain, which is a very sensitive organ. So this is defined often as either paradoxical inflammatory syndromes, of which one is called immune reconstitution inflammatory syndrome, or IRIS, when immunocompromised people become reconstituted and develop this very strong response, or else post-infection inflammatory response syndrome in non-HIV patients with have the immune system, and then they develop these issues, which often develop despite good response to antifungals. These responses are characterized by dominant pro-inflammatory Th1 type response. There is a lot of T cell and myeloid cell infiltration that can be tracked in the CSF fluid, and it is steroid responsive. So inflammation can critically contribute to disease pathology, and we are studying this aspect of cryptococcal meningitis. What kind of inflammation and how we can offset it. So we developed this model of IV infection with strain 52D, which is also now denoted as Cryptococcus neoformans. And this mouse model has this kinetic shown here in panel A, where we see this rapid increase of fungal burden in the brain, and then it starts going away. Now, what is interesting about this model is that we don't see any mortality during this upward expansion of the fungal burden, but we start to see mortality when the crypto starts going away. And this is associated with the severe infiltration with inflammatory cells and corresponds with this onset of mortality. So what happens in the brain sort of globally? We did this nanostring analysis of the neurodegeneration relevant genes, and what we see in this RNA transcription studies is that all the factors associated with the neuronal health, neurotransmission, and neuronal connectivity go down, while all those pathways associated with inflammations go up, especially cytokines, oxidative stress, and apoptosis, and other type of stress response that we expect to see in the distressed brain. So what type of inflammatory cells come into the brain? Very similar to what's been reported in human CFS. These are mostly T cells and monocyte derived cells, inflammatory monocytes, and then which then become MODCs. Of course, we have microglia in the brain, which are also an important part of the host response, but you can see this dramatic shift in proportion where in the naive mouse, the microglia outnumber the leukocytes 10-1, and then at the peak of inflammation at day 21, this proportion basically flips, where we have 7 times more leukocytes than microglia, despite the fact that actually microglia expanded as well. So, as I mentioned, we see a lot of T cells and both CD4 and CD8s, but vast majority of these cells are T cells. And these T cells are all Th1 polarized. We coined the name ultra Th1 polarized because we basically don't see any Th17 or Th2 cells, very few, while 90 something percent, sometimes 80, but always close to that number are interferon gamma producing cells. And these CD4 T cells are needed both for fungal clearance, but also contribute to mortality during CNS infection. So what we see here is the rigorous depletion of T cells with anti-CD4, multiple doses to make sure that they are not coming into the brain. And in this situation, despite the fact that mice no longer clear the infection and crypto continues to grow, we see 100% survival within 35 days, and we see that the murine, coma, and behavioral score doesn't go down. So we can preserve a lot of brain functions by depletion of T cells, but unfortunately that equals the uncontrolled expansion of the fungus. So in summary of this part, during this IV infection with strain 52D in C57 black 6 mice, we see this highly lethal meningoencephalitis in which mortality is linked to the inflammation rather than to the fungal growth, and that antigen experience interferon gamma producing T cells accumulate in the infected CNS along with monocytes, which rapidly mature into MODCs. These cells contribute to pathology And therefore, we conclude that immunosuppressive intervention can limit CNS injury, but the complete CD4 T-cell deletion would not be an appropriate way due to the fungal expansion. So can we somehow titrate down this inflammatory response? We looked at the inflammatory mediators that are being made in the brain, and what we see is basically pathways that are related to recruitment of the Th1 response and recruitment of Th1 cells, and also pathways related to recruitment of monocytes. And we focused on these two pathways first to see whether we can get some benefit to the host. And these pathways also are detected in people with cryptococcal meningitis. Here we see some iris response, and we can see that this MCP1, a ligand of CCR2, is one of the highly upregulated molecules in CFS and CSF, and we see that this is worse in iris patients. And here we can see in patient with peers, in our NIH patient, that vast majority of the T cells in both CD8 and CD4 are high in the CXCR3 expression, which is the ligand for IP10. So we started with deleting these molecules of interest. One is the CXCR3 receptor, which again senses CXCL10 and CXCL9. And what we see here is that the mice survive much better, then they have better behavioral score, and interestingly, they still are capable of suppressing the crypto. We also see in the wild-type mice this really strong cleaved caspase-3 signal in the brain, indicative of neuronal apoptosis. You can see the neuronal structures here that are all bright red, and this is much suppressed in the CXCR3 knockout mice. The other green color is the neuronal marker, which is called beta-3 tubulin. And you can see when crypto expands in the brain, it tends to sort of push aside the neurons, but just moving them around doesn't induce that apoptosis. It is this inflammatory response that we see in the brain. And what happens with the CXCR3 deletion, shown here with the single cell analysis, is that we are able to significantly reduce cytotoxic T cell surface molecules and reduce the markers of cytotoxic response against the target cells. So at least one of the pathways that we plan to explore in the future is the cytotoxicity of these CXCR3 positive T cells or other cells that accompany them. Now, how about CCR2? We also saw a great therapeutic benefit of the CCR2 deletion. We saw that this CCR2 deleted mice had much less of these negative transcriptional responses we see in the wild-type mice. So much less apoptosis, much less oxidative stress, and in many ways they more resemble the naive mice than the wild type infected with crypto. And here we see this beautiful response of beautiful marker synaptotagmin, which is one of the neuroconnectivity marker, where we can see a very strong signal around the cryptococcal lesion in the CCR2 deleted mice, while the synaptotagmin is significantly depleted in the area adjacent to the cryptococcal lesion. And here this is quantified. Basically, the MFI is almost the same as in the uninfected brain areas. So the effect is beautiful, and we were thinking about exploring it therapeutically, but unfortunately, with the CCR2 deletion, we also see a very significant suppression of fungal clearance. And this, if we would consider it as a therapy, it would have to be accompanied with, of course, antifungal drugs. So just to make sure we didn't overlook anything else, we looked at the strengths of the signals using the cell chat over time. This is a single cell analysis, and basically we are measuring strengths of incoming and outgoing interactions. And what you can see in the uninfected mice, that while macrophages are there and they kind of try to listen, basically most of the signals come from the resident cells. However, as the infection progresses, especially in the peak of the inflammatory response, the two chief cells that communicate become T cells and macrophages, which again tells us that these are the major cells that both drive the antifungal responses, but also this devastating brain inflammation. So to summarize what I told you up to this point is we have this meningitis in which two important components, CXCR3 positive and CCR2 cells, are important in inducing the damage and neuronal apoptosis, of which, you know, the CXCR3 positive T cell is definitely the bad guy, not needed for fungal clearance, but causing a lot of damage. And then we have CCR2 monocytes, which try to do a lot of good, but also induce a lot of collateral damage, perhaps by too erratic of the response. So do we have a IODA cell type in the CNS? We don't know, but the one that comes closest to this sort of positive hero is the regulatory T cell. They accumulate in the CNS along with the effector T cells, but they are somewhat delayed, so they don't peak at day 21, they peak at day 35. And this is sort of time when they arrive where we no longer see mortality in our mice infected who survived up to this point. So when we deplete Tregs, what we see is, of course, a significant worsening. The mice rapidly lose the weight, they become non-responsive, and they die very quickly, and all of them die by 20 days. And they die despite the fact that they clear the fungus even faster than the unmanipulated mice. And again, we can see exaggerated signature with the suppression of neuronal functions and enhance neuroinflammation and neuronal damage molecules. What happens is, which we can see it very clearly here around the lesion, these Treg depleted mice have much stronger infiltrates with the T cells coming and swarming around this cryptococcal lesion, much worse apoptosis quantified here, and overall bigger damage. So what we asked is, what is the mechanism? How Tregs can help in the brain? And one of the molecules that came first to mind was interleukin-10. And as you can see here, regulatory T cell is basically the major producer of IL-10 in the brain. So we use the IL-10 knockouts as our first approach to see if they can phenocopy the Treg cell depletion, and they do. We can see more inflammation, more T cells in the brain of the infected mice. We can see more cleaved caspase 3 and neuronal apoptosis in the brain, and basically every single parameter that we look at in these IL-10 knockout mice reproduces what we see with Treg depletion. And the final few slides that I want to show you is the possibility to use the Treg enhancement as a treatment in cryptococcal meningitis. This can be achieved by injection of mice with IL-2 complex, and as you can see, this leads to increased frequencies of FAXP3 T cells in the brain. And what is the consequence of this treatment? We see that CD4 total number goes down. They make less interferon gamma and less Tbet, so they are less activated and less of this ultra Th1 phenotype. What we see also is much less of this synaptotagmin depletion around the lesion, and as you can imagine, much less apoptosis after we treated the mice with the IL-2 complex. So the Treg immunotherapy substantially improves outcomes in CNS. You can see that we don't observe weight loss. They survive much better in that 35-day windows. Their behavioral score doesn't go down as much, but the trade-off is that unfortunately we see increased brain CFU. So again, we would have to, if we plan to develop this into therapy, we will have to make it under the control of antifungal drugs. So we see that Tregs have the protective potential, but possibly also therapeutic potential in protecting brain from the inflammation during CM. So in conclusion today, I want to highlight that during this highly inflammatory CM, Th1 type inflammation meant to clear the infection, in fact, drives neuronal damage and apoptosis. CXCR3 positive T cell subset is dispensable for fungal clearance, but drives both neuroinflammation and neuronal damage, and therefore anti-CXCR3 therapy would be beneficial. And in fact, we are working on developing one. CCR2 is also important for fungal clearance, but it also drives neuronal damage and by promoting this Th1-driven inflammation. And while Tregs are protective against CNS injury, and they most likely work through the anti-inflammatory IL-10 cytokine. And immunotherapy expanding Tregs is highly protective. However, we need to mind the fungal control. So together, our data imply that targeted interventions that modulate specific inflammatory pathways in CM can significantly affect the clinical outcomes and should be explored as complementary therapies, especially in PIRS and IRIS. There are many people I want to acknowledge, but most of all, Jintao Xu, who works on this project for many, many years. And most of the data I showed are either published or soon will be published in papers he helped to prepare. Hailong Li, who work on the Treg and IL-10 project. Laurie Neal, who was the first one who actually helped us to develop this model of 52D infection. Christy Gaggenauer, who always pitch in to help whenever we have those humongous harvests. She has her own project now, but through being so close with us, she's always there who help. And two technicians who helped us to breed tons of mice and harvest them. Big thanks to Rylan, who did a lot of IL-10 work independently. Finally, we have the big army of undergraduate students. I only listed those who either recently graduated or are still in the lab and contributed greatly to our work. I want to acknowledge my collaborators from NIH, Peter Williamson and Jessica, for giving us the human data and many great ideas, and also Clifford's lab for help with the microscopy. Thank you for your attention, and I am looking forward to your questions. Thank you.
Thank you very much, Michal, for a very excellent talk highlighting the difficulties that are faced in trying to deal with such a complex immunological problem. Okay, well, thank you, both speakers. Thank you very much for your talks, and we will start with the questions. And now we'll start by posing three questions to Bernd. So the first question is about the HIV status of your patients and essentially whether HIV has any impact on the outcome of the alcoholic liver disease, Bernd.
Yeah, very important question. So for us, we did our exclusion criteria are actually HIV positive patients. Just we are not able to work with these blood samples and stool samples. So just for logistic reasons, we did not include patients with HIV and alcohol use disorder. But obviously, this will be a very, very important question to answer. If these patients who are immune compromised, would they have actually more Candida albicans or in general more fungi? And because we know that patients, especially with advanced liver disease like cirrhosis or alcoholic hepatitis, they have this very weird they have this very high inflammatory response and systemic inflammation. On the other hand, they're very immune compromised already by itself, by whatever means. And we call this immune paralysis associated with cirrhosis. So there is probably something inherent already like in these patients that they don't even need HIV to be further immune compromised probably to get to this stage of immune suppression, and then you get this overgrowth of fungi and Candida albicans.
Okay, thank you. So there was a question around the anti-Candida specific IgGs that you see in your patients, right? And it's known that Candida colonization leads to the generation of antibodies that target cell wall as well as Candida lysins. The question is, in your patients which have these higher anti-Candida IgGs, right? And you've shown that beta-glucan and Candida lysins contributes to damage, and presumably the antibodies targeting these components would sequester them away and prevent them from causing damage. So do you think these antibodies targeting these components, beta-glucan, Candida lysin, are less in alcoholic liver disease patients, or not functioning, or not blocking? What is your thoughts?
Very good question. I see this, and this is more or less really speculation because we have not really done any real experiments, proving this. But my speculation is that this is just a protective mechanism, and you get like more, gut leakiness, you get more translocation of these fungal products to the systemic circulation. So I think this is probably a response from the body to just this more, to this leakier gut, to this higher burden of fungal products like in the systemic circulation. Now we tried to measure anti-Candida lysin specific antibodies in our patients. We unfortunately were not able to succeed we tried this many years ago. Maybe there are actually better antibodies out there now, but we got one and we could not really, with recombinant Candida lysin was not a problem to get good ELISAs established. Right. But then when we tried to really measure, for example, mice that were gavaged with the Candida, or we know there is Candida lysin present or not in the serum, even if we spike it in, we could not detect it. So there was a limitation with the ELISA. And honestly, we have also not very good experience with the ELISA from the beta-glucan, this is the other way around. So we wanted to look, we have never looked actually at beta-glucan antibodies in our patients, but this would be also certainly informative, but I suspect that they will very, very well behave like, similarly like the Candida albicans IgG, that it's going up during active drinking, and when we have all of this ongoing translocation, and then once this is stopped and once the patient stops drinking, this might also go down. I don't know how rapidly, but I think that's my suspicion. So technical limitations really prevent us to look into this more detailed questions.
Yeah, I think that the question was also aimed at whether the antibodies would modulate the ability of those two agonists, liver agonists, to function, but you haven't used
right. Yes. So we are trying this right now with a different antibody, but we are not targeting them systemically. We are targeting them actually just enterically. But let's see so we are just starting this experiment, but let's see how if that shows anything.
Okay, my last question is also related to Candida lysins, about the concentrations that you need to cause damage, and whether you think there's a sufficient amount of Candida lysin getting out of the gut and causing damage in the liver.
Yeah, I have to admit, I mean, this, and this was obviously also like, that's why we also wanted to measure like anti-Candida lysin antibodies in the patients, but also in our mouse model. We have also not been able to directly measure Candida lysin in the serum, for example, from mice, also due to this limitation that we don't have a good antibody. We have not tried mass spec, which would be a possibility, I guess. But so this is more or less a little bit, I presented it that we assume that Candida lysin is higher in the portal vein and possibly in the systemic circulation and in the liver. But really, this is our speculation. Right now we have not been able to really show that Candida lysine, the peptide, is really higher in the portal vein or not. Yeah, so clearly there is this missing link, and I completely agree. That this is some area that we still need to further focus on, and hopefully with better tools, like in the future, we can really prove that.
Okay, thank you, Bert. Over to you, Geraldine.
Okay. Do you know if the Tregs generated in your model are specifically targeting the ultra Th1 cells, or are they generally suppressing the total effector response?
Yes, that's a great question. So the answer is yes to both part of this question, because yes, we see a very massive suppression of T cells in terms of what comes in the brain. So when you globally look by who was affected, we see that basically virtually all subsets of T cells go down. However, the most reduction we see in those Th1 cells, so they've been curbed the most. But then when we start looking into the subsets like Th17 and Th2, which we see a little bit, now they are more overrepresented in the gemisch, so to speak, but they are still fewer of them than we would see in the fully blown infection. So they go down by a little bit. So yes, answer is yes for both. We predominantly see the ultra Th1 effect, but also the other cell subsets are to some degree affected. Thank you.
Okay. And another question. So what emerged from your talk is that the ideal treatment would be to deplete CD4 T cells as well as treatment with something like amphotericin B. So have you tried that in mice? And is the effective dose lower?
We have that. And, and this is the sort of like when we did this sort of 2x2 design, so T cell depletion alone, T cell depletion with amphotericin alone, and doing nothing. So from the perspective of weight loss and the survival time, the best group was the combined therapy. But eventually, all mice get very sick. After 70 days, we're gonna lose them all. The amphotericin that we use didn't penetrate very well into the brain because we haven't used the liposomal ones. So even though this group was the best, we didn't see the effect. So now we are looking into different combinations. We are trying to use Ambizome, but most of all, we are trying to look at the depletion of the CXCR3 positive T cells, because they contribute the most to the damage and are have very little contribution to fungal clearance or are dispensable in some of the scenarios. So these studies are ongoing, and hopefully in 6 months or so we'll be ready to submit them for publication.
Okay, thanks.
Yeah.
Back to you, Gordon.
Thanks there, Geraldine. So, Birds, I have a talk that's also related to amphotericin B, but and that is whether you've tried other antifungals in your model and if they work equivalently. You're on mute, Bernd, sorry.
Sorry. Yes. We actually, interestingly, we started off with fluconazole. And then fluconazole, as you know, it's being absorbed. So, and then all of a sudden, in our very first experiment, we actually saw all of a sudden liver damage, and we know this from our patients, you know, that common side effects, that all of a sudden we saw liver damage in our control mice. So that all of a sudden they had liver injury and steatosis, and then we definitely knew we had like a problem. So this, you know, clearly had some liver toxic effects. We then switched to amphotericin B. This is not the liposomal, just the regular amphotericin B orally. We do not anticipate that actually this goes to the systemic circulation, so we would not have like any we did not see any toxic effect. So this was our first paper that we published. But then I also was suspicious, you know, is this really you know, can we use other antifungals? And in follow-up studies, we have actually published now 2 papers with nystatin, very similar, you know, orally available, gut restricted, not going to systemic circulation. And we see the very same effect like with amphotericin B, and also no liver toxic effect. So we have, you know, with nystatin and amphotericin B orally, we see the same effect like on the liver outcome and on Th17 response.
Okay, and then talking about the protection, but from a different perspective. So when you used your ECE1 mutant, you could show that you were able to, essentially, the mice didn't show the same level of liver disease. And the question is, could this be like a vaccination? Could you treat the mice if you came back then with wild type candidate? Were the mice then protected? Is there some sort of T cell memory that can kick in or something like that?
Yeah, that's excellent point. We have not tried that, so we have not challenged them or primed them, and then we just gave them ethanol, or you know, re-stimulated them, or not re-stimulate, but essentially after stimulation we did not look after a while, you know, for the response to alcohol. So we have not tried this, but I could imagine that there might be, you know, adaptive immunity kicking in that might be protective eventually, you know, against some of the, you know, for example, cannula lysine peptides, for example. So that's a good point that could be certainly studied in the future.
And then my last question before I hand back is the link between the microbiome. So you've shown that very clearly for alcoholic liver disease. What about like viral disease, like hepatitis or other viral diseases? Do you see a similar link there? That fungi impact the pathology of those diseases?
Interesting point. So we have in our if we do the same, so we essentially have, you know, in terms of liver diseases, we have the 2 major liver diseases, you know, is in the Western countries. In China, it's a little bit different. We have more viral hepatitis. But here in the US and also in Europe, we have, you know, alcohol, and we have this new nomenclature now with metabolic associated, dysfunction associated steatotic liver disease, MASLD. Before it was called non-alcoholic fatty liver disease, or NAFLD. And we have also a very large cohort of patients in my laboratory. And for example, we also looked at fecal, you know, if we can grow fungi actually from these patients, and there was no growth at all. I mean, it's very similar to control patients. We rarely can culture, like from our control patients, any fungi from the feces. And these are just regular YPT plates, and nothing you know, spectacular plates or very sophisticated culturing techniques. However, when we also treat mouse models, and in this case, we actually used germ-free mice, we colonized them with stool from patients with MASLD. And then if we use, for example, amphotericin B, we also see a significant improvement in experimental induced or diet-induced steatohepatitis and liver fibrosis in this case. But we have not done anything in viral diseases.
Okay, thank you. Back to you, Geraldine.
Okay, one very general question. So your talk focused on Cryptococcus neoformans. Do other Cryptococcus species have the same effect?
We worked with two cryptococcal species, C. neoformans and C. neoformans, which are still C. neoformans species complex. So they still probably are all diagnosed as C. neoformans. I haven't tried to infect brain with C. gadi. I know it is much less prone to cause brain infection, although sometimes the cases are seen. So for now, our focus is on C. neoformans species complex, but we are looking more into both peers and non-peers situation, because we still think even in patients who do not develop peers or iris, there is likely inflammatory component that is simply bad. And if we can tease it out and take those bad parts of inflammation away and leave the ones that are necessary for clearance, I think it may benefit also non-iris or non-peers patients. So we start looking at this. There are some interesting data, but too early to talk about it at this point.
Okay, so were you addressing there the last question about how to better identify patients at high risk for peers?
Okay, so large body of work from Minnesota group, and in collaboration with Kampala, with Uganda, they identify a lot of factors that predispose to iris infection. Now, peers, it's much rarer occurrence, because fewer people who are non-HIV or transplant patients develop this spontaneous cryptococcal disease, and usually they are treated by standard antifungal treatment. So if they are not to develop peers, we don't know about those patients. We don't have the readouts. The only reason we know somebody has peers is because they are refractory to antifungal treatment. So I think there's still some work ahead to see were there any readouts in these severely compromised patients or previously healthy patients that would tell us they may develop peers. And of course, we don't know, because by the time we see them and the NIH sees them, they often are already crypto negative. They already cleared. It's just this lingering inflammation that continues to erode their brain. So, so yes, that's a great question. I don't know the answer to it yet.
And what about in the mice? Can you identify anything in the mice that would help you predict?
Yes. So the two models that we use are different, but it's a good idea to compare the peers model and non-peers model. The issue is that the strains that we use for the peers model and non-peers model are so completely different in virulence and in their properties that the conclusion why they are different will not be so easy to achieve. So we are looking into this, maybe using different mice strain. Still, we didn't look at that early time point, but we see that in one of the models that we did some preliminary data with the same strain 52D, the mice don't develop PIRS and they don't develop the ultra Th1 response in the brain as well. But we need to look a little bit more into that to tease it out. So future direction, great question. We think of it, but no data yet. Thank you.
Okay, thanks. Okay, Gordon.
Right, thanks, Geraldine. So we've had a comment feedback from Bernie Huge about Bernd's reply about candidiasin in the blood and detecting it. And according to Bernie, this is likely to be very difficult because candidiasin becomes very tightly bound to proteins and other molecules in the serum. So thank you, Bernie Huge, for that one. Bernd, so a question here about the microbiology that's happening in the gut. So is the proliferation of Candida due to the alcohol-induced metabolites or the suppression of the bacteria in the gut?
Yeah, that's an excellent question. That's what we are chasing, like since we made this discovery, you know, that we see this fungal overgrowth after alcohol feeding, like in our mice and also in our patients. We haven't really gotten to a point where we can with certainty that we can say, you know, there is either alcohol metabolism plays a role, microbiome changes that would allow the Candida albicans to overgrow, or is it from, you know, the effect, like I said before, on the immune system from the host through alcohol, chronic alcohol abuse, where we know that we have, for example, you know, less immune response to translocated bacteria, fungi, probably in the gut itself. So these are all possibilities, and you know, but we have looked into so many different pathways and possibilities, but have not found a very conclusive answer. So certainly, you know, bacterial microbiome changes upon alcohol, they might facilitate, like, you know, overgrowth of Candida, may make a you know, very nice environment, ecosystem for the Candida to grow.
Okay, thank you. So I'm going to use my chair's prerogative and ask two more questions, one of you, Bernd, and one of you, Michal. So you mentioned germ-free mice, Bernd. And the question that I had when you were giving your talk, so if you give lots of alcohol to germ-free mice, are they protected significantly from the developing alcoholic liver disease?
No, we tried this very, very early on, and you know the problems with the germ-free mice, right, itself. They have a completely dysregulated immune system, and one of the, and this was like a very early paper on, like when, you know, the gnotobiotic and germ-free mouse came up, like 2000, probably 20 years ago, when they compared germ-free mice and conventional mice and did for, RNA sequencing in the liver, one of the highest dysregulated proteins was the CYP2E1, so cytochrome P450-2E1 that metabolizes alcohol. This is much, much higher in germ-free mice. So you probably need some, and we know this also, like for TLR4, for example, it's also higher, right? So you have this dysregulated alcohol metabolizing enzyme in the liver, and if you give actually alcohol, and we did this very early on, we gave them like just one binge of alcohol drinking. And then if you harvest them like after 9 hours, the alcohol metabolism, so the alcohol itself is much higher metabolized. So the blood alcohol level is lower in the germ-free mice. And all of a sudden, because it's more metabolized, because you get more reactive oxygen species, you make more liver damage. And ALT is much, much higher. Steatosis in this model is not a very good readout, but you get actually more liver disease in the completely germ-free mice. So you have a component, as we know, you know, that regulates some of the immune system, and in our case, you know, this very important alcohol metabolizing enzyme. But that's a good question.
Thank you very much, Bernd. And then my last question to you, Michal, is one of the things that struck me about your models was that you get these really high cryptococcal burdens in the brain and no obvious phenotype terms of pathophysiology, at least in terms of your mental scoring and other parameters. So how high can you go before you actually see something happen to the mice in terms of fungal burdens in the brain?
Yes, I think it also depends which strain is used for the infection. So for the C. neoformans 52D or CD neoformans, we go above 8 log, so 8.5 log before we see some issues, and you know, in the absence of inflammation, those issues are much delayed. In the presence of inflammation, we see them much earlier, because of course of those effects. So we're gonna see the head bump or the evidence of increased intracranial pressure. We'll see some bleeding in the brain, and of course microscopically we see tons of neuronal apoptosis. So these happened sort of independently of the fungal burden. Now with the H99, which is a highly virulent strain, we will see some of the symptoms when the fungi reach 10 to the 6th and to the 7th level, and this model is much less investigated, but they also have inflammatory component. So we are also looking to what's going to happen to these mice. But what is interesting with this H99 model, when we deplete CD4 T cells, just like we deplete them with 52D, the mice actually die tiny bit earlier. So in this case, the CFU is also an important driver of the response, but there is also an innate component that we start to look at. So a lot still to learn about, you know, the interactions between different strains of crypto in the brain, and also different types of inflammation they trigger. So
yeah, that's great.
Good question. Good question.
And the fact that you've got these two different strains, it's going to give you lots of insight. So thank you for that.
Yes, yes.
And thank you to our two speakers. We just passed the 5:30 in the UK time. So thank you again to our two speakers for really stimulating talks. Thank you to my co-chair Geraldine, and thank you all for your attendance. And these talks will be up, I think, online next week. And I'll wish you goodbye. Thank you, everyone. Bye-bye.
Thank you. Bye-bye.